All right, welcome back. Part 2 of 2017. Higher level paper 2. So nucleosome, those are when you have these histone proteins and you have DNA wrapped around them. Usually you have 8. And there's 4 in the background. So that's a nucleosome. I think I pulled this question and used it for an exam earlier this year. So the chemical modification that could impact gene expression in this nucleosome. You have methylation, where you have methyl groups added, CH4, acetylation, phosphorylation, and those all have an effect of bonding certain areas of the gene together and preventing expression, or at least slowing it down. So those are some chemical modifications. Probably methylation we hit on most, so that's something you really want to focus on. Main thing to remember, methylation turns genes off, acetylation turns genes on. Okay, so they acetylate, they will be turned on. They methylate, usually it turns them off. So here we have a tRNA X-ray crystallography, exactly the technique that was used to discover the structure of DNA. And there's 2 binding sites. And this one right here is where you have the amino acid. And down here is where you have the anticodon. So you might have UUA, getting ready to bond. The AAU on the messenger RNA, on the mRNA. So the question here, outline the function of the 2 binding areas. So binding 1, area 1 carries an amino acid. And when it comes to the ribosome, it'll leave that amino acid on that growing polypeptide chain. And area number 2, that's the anticodon, and it binds the codon, binds to the codon on the RNA. Remember, the codon are those 3 base letter groups on the messenger RNA. 3 codons for each amino acid. Remember, 3 codons, 3 bases, meaning A, U, C, or G when we're looking at RNA. So free and bound ribosomes, free, just floating in the cytoplasm, just floating around in the cytoplasm. Bound, those are going to be on the ER. So anything, usually proteins made on the free ribosomes, are going to stay inside the cell to be used by that cell. Proteins made on those bound ribosomes typically will be exported. They'll go through the ER membrane structures and then go through the Golgi and then get released, usually through exocytosis, to go on to other parts of other cells and other areas that organism needs. So here is the classic Louis Pasteur experiment with using all these different types of flasks to basically figure out what's going on with this concept of spontaneous generation. Where does life come from? A lot of people really thought that life just popped out of nowhere, that a bunch of dirty rags would create mice, or just poop. Flies will just pop right out of poop. So it took, and we all know how that happens now, but it really was a question 200 years ago. Actually, here's 1922. Okay, just 100 years ago. So, but what Pasteur found, that the flasks that were cut off from the outside air, no bacterial growth was observed. Now, remember, he used a broth in these flasks. The broth was boiled before the experiment to kill any previous microorganisms. So any existing and any flask that was cut off from the outside air, nothing grew. Okay, it was only ones that where air could get in that started to develop signs of bacterial growth, cloudiness, nastiness, funk, whatever. So yeah, the flasks that were cut off from the outside air, flasks that were not cut off from the outside area, bacterial growth occurred. All bacteria were killed in all groups by boiling it before the experiment. Yeah. So basically, if there is a source for new bacteria, you saw bacterial growth. But if it was cut off from any other stuff from getting in, then you didn't have any bacterial growth. Showed that broth wasn't what did it; it was whatever was getting in. There was something in the air that was bringing some kind of organism in to start growing. Okay, so paramecium, that's a unicellular organism. You know, from pond water. We didn't see any from the LA River water. But cilia, movement, locomotion, or feeding, nutrition. Remember, you just have one. Do not, on your exams, do not write a whole bunch of things and just hope that they're going to pick the correct one. They're going to read the first. So the examiners will only count the first thing you say. The first, say the first word or idea. See, it's 1 point. They're just going to look for that first thing you write. So if you state the function, if you have the wrong function in your first one, but you have the correct one in your second one, they're not going to count the second one. It'll just be wrong. So you just can't throw out a bunch of stuff and have them pick the one that's correct. Make sure to choose one and go for it. It's kind of scary, but just got to do it. The contractile vacuole, basically it's a way for the paramecium to pump extra water out of the cell, so that's important for homeostasis, osmotic balance. Yeah, doesn't want to burst, basically. Remember, in fresh water, this is a freshwater organism. Water's constantly coming into the organism because it has a higher salt sugar concentration. And the opposite is true of any animal living in the ocean. In the ocean, it's always, losing water to the environment. So it's got to always be drinking. It's not worried about getting rid of water. That's happening all the time. So it needs fishes in the ocean that got to drink a lot. And they don't pee much. But freshwater fish pee all the time. They're trying to get rid of that water. All right. Adult stem cells. Great because stem cells in general can divide over and over again and become different kinds of cells used for treatment. Can't be rejected, since, like, if I needed adult stem cell treatment and I got my own stem cells, I can't reject them because they're mine. And there have been some really scary, mysterious cases of cancer being started by the use of stem cells from one person to another. So reducing that chance of being getting cancer is important. And there's fewer ethical issues. Remember, the main issue is embryonic stem cells, right? Those are the ones that people freak out about. People think there's some people believe at conception that's when the life begins. So anything that happens to any embryo is like killing a full human being, which really is not the case, because if that were the case, remember, we're keeping human beings frozen in freezers all across the world And then those humans are being thrown in the garbage if you can't pay the storage fees in the freezer. So all the right to life people never talk about that. That's what they should be protesting, in my opinion, because that's pretty messed up, keeping human beings frozen in a freezer. So the reason why embryonic stem cells are used, even given those supposed ethical concerns, they are ethical concerns for some people. Don't want to discount that. The reason why embryonic stem cells are preferred because they're more effective. So that's the disadvantage of adult stem cells. They're not quite as pluripotent. They don't quite develop into as many different types of cells or as effective. And they're pretty painful to obtain these. To obtain these cells, you gotta go into the bones and suck them out, and that can be a pretty painful process. Okay, so adult stem cells, good. Fewer ethical issues, no rejection, less cancer, but not quite as effective and painful to get. All right. So let's see here. Oh, there we go. Big problem that researchers are worried about as far as global warming, climate change, is all this carbon dioxide that's being released as Arctic soil thaws out. That permafrost goes away and becomes a muddy mess, and therefore any organic material is going to start to decompose. So this is showing a rate of release, ecosystem respiration rates, as the soil temperatures climb. So this is warmer, this is colder, right? And you can see a lot more respiration as we see these warmer temperatures. Okay, above. Remember, this is freezing. This isn't Fahrenheit, so this is freezing, the freezing point. So if it's below freezing, it's pretty low, right? But once you go above freezing, then it starts to pick up. Although here, it's interesting. This is below, a little bit frozen, but you still have some bacterial action beginning. So the effect of increasing carbon dioxide release on global climate change is going to increase the enhanced greenhouse effect, and more CO2 is released due to human activity, basically fossil fuel burning. That's what we're talking about. The one thing we as humans do, we burn a lot of fossil fuels. We just need to figure out a way to get away from that. So most of the surface has a wide diversity of ecosystems. Two general characteristics of all ecosystems. So this is a super broad. They have biotic and abiotic factors that interact to sustain life. So remember, biotic is anything living. So the plants, the animals, the microorganisms, fungi, etc. And abiotic factors like water, sunlight, weather phenomena, rocks, things like pH. Those are all abiotic factors. Nutrients need to be recycled in all ecosystems. Energy flows in and out. Remember, energy is not recycled, but nutrients are recycled. Remember that distinction. They might ask about that. And in all ecosystems, you absolutely need those decomposers because of this point right here, that nutrients. You can't recycle nutrients if you don't have decomposers. Alright, vascular plants. So those are more complex plants like conifers and angiosperms, where you have phloem. So coniferophyta and Angiospermophyta. So outline active transport. So it's used to load sugars in the companion cells, in the phloem cells, and hydrogen is moved out of the phloem to make a hydrogen gradient. Once that happens, then sugars can flow into the phloem sieve tube members. So sugars, amino acids, so remember, using ATP, I'll also put that point. And how does it replace water it loses in transpiration? Yeah, these are questions on paper too. Creates that suction tension in the water column in the plants. Xylem tissues, that helps it kind of give a pull because of the cohesive and adhesive properties of water, primarily cohesive. In that case, water enters the roots via osmosis because there's a higher solute concentration in the root tissue, and water can kind of grab onto itself and other water molecules using the cohesion, and then it sticks to the xylem using adhesion. Okay, remember, it could be low pressure. That means, the rigid walls of the xylem are necessary. All right, so let me go ahead and pause there, and then we can do section B.